Hi friends, myself Dr. Sonu Panwar, and today I am discussing up the AIIMS June 2020 microbiology questions. And obviously I could not able to get all the choices correctly, so I am not saying that I have all the options which are correct ones here. So the idea is that you should get an overview of that particular topic. In and around that, if at all the question next time comes up, you will be able to answer up. Okay. And yes, I could not able to get much of the options, and if you know it, then you can put it in the YouTube section, and that is in the comment section, and we can discuss of that particular thing later. Okay, so first of all, I'm starting up, and the first one is this question: all the following are measures used against the COVID-19 except. So obviously nothing is 1% glutathione, it might be 2% glutathione, but it is not used for the COVID-19. That everybody knows. Coronavirus disease, there is no use of 2% glutathione. You use what? You use 70% ethanol, you use face mask, and you use hand wash. Okay, these all things are used. You know, proper hand wash is mandatory, face mask wearing is mandatory, 70% ethanol is used as sanitizer. Along with that, 1% sodium hypochlorite is used as a sanitizer. Okay, but 2% glutathione does not have any role, so that should be the answer against this one. The first answer is A. Second answer is biosafety level of SARS coronavirus 2. This particular one, which have caused COVID-19, obviously, that was asked that what is the biosafety level of SARS coronavirus 2. So for this particular question, if again it was like a divided view, some were saying that sir it was just this, that which level is used for this, and some were saying that for culture and isolation which particular level is used. So this is there in my YouTube video, and last lines if you observe up in that, I've already told that if you're doing up a RNA extraction and doing the PCR with the RDRP gene and all, then the level needed is what, 2, and if it is for the culture and isolation, the level needed is 3. So BSL2, BSL3 both can be the answer, but depends upon what is the question asked in the exam. And again, the students are confused up. They could not able to tell me that is it the culture and isolation. If it is culture and isolation, BSL3, and if it is a routine working with the this particular SARS coronavirus 2, that is the BSL2. Fine. Then the antibody testing for coronavirus 2 depends upon this another controversial question. They could not able to give me the proper choices of it, but the only thing which seems relevant in this is it tells about the duration of disease. Okay, if you're going for IgM antibody testing, it will tell about the duration of disease. If you're going for IgG antibody testing, it will also tell about the duration of disease. Fine. So it tells about the duration of the disease. Okay, antibody testing coronavirus depends upon this duration of disease. It can tell IgM and it can tell IgG. Fine. I don't know that these were the options there or not. Currently infected and asymptomatic. Yes, this can also happen up. If it is given up that IgM antibody testing is done for what? So IgM antibody testing is, then the answer might be this: currently infected and asymptomatic. Okay, the person is infected, but the IgM is not coming up, or you want to see up that IgM have come and the symptom and signs have not come into that particular fellow coronavirus disease. So depends again on the choices, what are the choices and all. Sorry to say, the students could not able to reproduce well in this, the various options. So answer to this question obviously tells the duration of disease. Duration of disease means it will be telling up, is it an early disease? So IgM will be positive. It is a late one, so IgG will be positive. PCR starts coming positive right from the start. Okay, the IgM comes at which particular time? It comes at around 7 days, and IgG starts coming up at around what, 14 to 15 days of this particular one. So again, you can go for as per this particular knowledge of the options and all, that what should be the answer. Okay, now this interpretation is there. Viral load can be estimated by what? The viral load, if this was a question there, so obviously the real answer is real-time PCR. But this particular, or any PCR, can give up the viral load stuff very easily, and it is estimated to the best of the levels. The viral load, if it all comes up, then the best answer would be this, because from the ELISA also you can have the load. Remember this. Okay, but answer to this particular question, if it is there, RT-PCR will be the best answer. Now this picture was there in the exam, that is the Sporotrichosis picture, and this particular one is the commonest variety of this one, that is the lymphocutaneous type. You know that Sporotrichosis is what? It's a dimorphic fungal disease, and if it is a dimorphic fungal disease, obviously it will have a yeast form as well as the mold form. Yeast form looks like a cigar-shaped yeast cell, and the mold form, flower-like sporulation. And this lymphocutaneous type, what is there, which is seen in this picture? The picture is showing up these types of lesions, and this is termed as which pattern? This is termed as sporotrichoid pattern of spread. Okay, and what you have in this? A nodule, and then what happens in that nodule? Ulceration. Okay, so obviously there was history of rose gardening and all, so it's also termed as what? Rose gardener's disease. So it's a straightforward question. Then another question comes about the sedimentation technique, that is the formal ether sedimentation method for fecal material. In that, that what you take the fecal material in which you want to find out the helminthic egg, and you do a series of things with it, like you put it into a gauze and you want to filter it off, and after that you can centrifuge it, after that you can add formalin, after that you add up ether, and after doing all these things, what you get, these particular various strata. The upper one is ether, then you come the fecal debris, then the formalin, and then sediments, and the ova is there, where in this particular fellow, if you label it as 4, this is 1, this is 2, this is 3, this is 4, the answer should be from where you will have the eggs and all, from the sediment. Okay, so that's where the name also comes, the formal ether sedimentation method. And like this, there are flotation methods also. Here you are taking sediment, and there in the flotation one, what you are taking up, you will take up the flotation method, you will take up the upper portion, because flotation means they will float up. What you have done, you have put the fecal material into where? Into a particular thing which is having a high specific gravity, and because of that, the eggs will float up to the upper surface, and that is what is termed as flotation methods. And two are there. One is normal saline flotation method, and another one is zinc sulfate flotation method. So the answer to this question, formal ether sedimentation method, is that sediments you will observe to find out the egg. Okay. Now, this was given up in the exam, that this is a sort of a sandwich type of ELISA, and okay, what you have in this, that this particular antibody is there, and this antigen is there, target antigen, which you want to find out, and another antibody is there to which this enzyme conjugate is attached. And what is the name of this? HRP. And this HRP conjugated detected antibodies is sandwiching this antigen in between, and then you add up a substrate, and you need a chromogen also, that is termed as TMB. So this is ELISA, and sort of a sandwich ELISA type, and because antigen is being sandwiched between the two antibodies. Fine. The names you can remember up, that HRP is what? This is termed as horse radish peroxidase. This is a commonly used enzyme conjugate. Another thing, the chromogen. The chromogen is an indicator which gives the color, and the example of chromogen just now in the picture also, that was TMB, but the commonly used one is OPDO, that is termed as orthophenyl diamide. Fine. Then the substrate name is this para nitrophenyl phosphate, and you can also use hydrogen peroxide as a substrate. So enzyme and substrate, this particular reaction is seen with the help of what? Chromogen. Okay, so that is what is termed as that type of ELISA, sandwich ELISA. If at all that picture was there, you can imagine up that was the conjugate which was being given up. This was there, the acid fast organisms of 4 to 6 micron. There is a history of renal transplant, and then they are telling the persistent diarrhea happens up. The most common species affecting the humans is Cryptosporidium parvum to cause diarrhea. That I don't know this was there or not. Again, confused version is there, but this size is characteristic of what? Cryptosporidium parvum oocyst, and which are founding in the diarrheal episodes. And remember, for this particular one, we use a drug termed as what? Nitazoxanide. Okay, and if you know the other options and all, might be the answer change. I don't know the exact options. I am going up with the size, that is 4 to 6 micron, and that is of the size of the oocyst of the Cryptosporidium parvum. Okay. Then isotype switching. Isotype switching happens what? Because of the change in the constant portion of the heavy chain. Okay, And these are the options: active B cells, cytotoxic T cells and B cells. So answer to this question is active B cells. Okay, whenever the activation of the B cell, when the B cell is stimulated, means activated by an antigen, then what happens? The heavy chain gene undergoes rearrangement, in which the variable portion, that is VDJ, that is the J is the joining one, D is the diversity, V is the variable portion, combines with the CH gene segments. Constant portion, heavy chain, segments it, combines up and comes out with the first antibody, that is the mu, IgM. That's why always the first antibody to come is what? IgM. And later on in this, the further change can happen up. It can change to IgG, it can change to IgE, it can change to IgA, or this can change to this can change to this. So that is what is termed as class switching. And class switching is a phenomena seen up where the class switching is a phenomena seen up when there is a change in the constant portion of heavy chain. Okay, and it happens with whom? It happens with the activated B cells. So that is what is the answer of it. Then the cell-mediated response is responsible for type 4 hypersensitivity. Everybody knows it, and this is the straightforward question. Then there was an image given up of a life cycle with a rodent, 6-leg larvae, 8-leg adult, transovarian transmission, and it was asked that which particular one transmits it. The answer is trombiculid mite. They are talking about which particular disease? They are talking about scrub typhus. And you know scrub typhus comes under which particular family? That is rickettsia, but it is different from rickettsia because it comes in the Orantia stuff. And scrub typhus is caused by what? Orantia susugamosi. That also you know. Okay, and this Orantia susugamosi, this particular organism is having this type of life cycle, that these are the eggs, and these eggs they change into what? 6-leg larvae, and this 6-leg larvae frequently affects the humans. So that is also named as what? Chiggers, and it is termed as chiggerosis. Okay, this particular thing, that's why it is termed as scrub typhus. Another name is what? Chiggerosis, because of this 6-leg larvae. Then this larvae feeds on the rodents, and what is formed up? This 6-leg larvae changes to what? 8-legged nymph and adults, and from the adults to the offsprings, what happens? Transovarian transmission, and there is a formation of egg. This is the life cycle of Orantia susugamosi. Another thing which you need to remember here is that this is OXK positive, and OXK positive means that is heterophile agglutination test, wheel Felix reaction, which is positive for what? Which is positive for this particular scrub typhus, and it is sort of a diagnostic. And remember, indirect immunofluorescent testing is done here, which is a gold standard. And in this, you got to see a tetrad, and that tetrad is named as what? Zoonotic tetrad. And what is a zoonotic tetrad? In which you will find mite, you will find mice, you will find a wet season stuff, and you will find the shrub vegetation. Shrub vegetation means small vegetations. If these 4 things are there, this is the ideal condition to get what? Scrub typhus. Okay, so that is what termed as what? Zoonotic tetrad. What is the drug of choice for this condition? Drug of choice is doxy. For all the rickettsial infections, what is the drug of choice? That is the doxycycline. Okay, so these all things are there. Then there was a question regarding the gene expert, and gene expert detects which gene? Everybody knows gene expert. RIF is a full name, which is a CB-NAT from the Cepheid. And remember, RPBO gene is the one which is being targeted. Okay, so answer to this question is third. Remember other names also like CATG, INHA and PNCA. They are also responsible for their respective drug resistance. Like CATG is for isoniazid, INHA is for again isoniazid, and PNC is for pyrazinamide. Okay, so these are the various resistance of it. Now this was this particular picture was given up. Obviously the leptospires was not written there, but it was a picture of what? Leptospirosis. And leptospirosis you already know from the notes and from the app. It is triple R. What is triple R? Rat, rice and rain. Okay, rat, rice and rain, and it leads to a disease particular termed as what? What is rat, rice and rain? That is rat urine, rainy season, and it happens to those who works in paddy fields, right? The rice watery fields. Okay, and thereby they get what? Fever. They get like icterus, and they can get number of other things as a feature like conjunctival suffusion is there. That this particular word you need to remember, conjunctival suffusion. What is conjunctival suffusion? Conjunctival suffusion is not conjunctivitis. it is red, but there is no inflammatory exudate seen in this particular disease. This can be a question. Okay. C, another hint, uveitis. Uveitis, autoimmune uveitis is a feature of leptospirosis. Remember this particular thing also. There are two phases here. One is leptospiremic phase and one is immune phase. The early phase is leptospiremic phase and the mid phase is immune phase, in which antibodies start coming up. And leptospiremic phase is a phase in which hematogenous spread happens. Okay. And obviously you know that particular thing, that this is the investigation we use for it, MAT, that is microscopic agglutination test. And nowadays investigation of choice is going towards what? PCR. And the drug of choice, if somebody asks you regarding leptospira, again it is doxy. Okay, so doxycycline is being used here. But yes, if the leptospira is very serious stuff, then you have to use what? You have to use the penicillins. Fine. Now, what should be the minimum dose of tetanus antitoxin in the body to provide the optimal protection? So, answer to this question, as per the PARC, is 0.01 international units per liter. Okay, so this is what is the answer here, because that was very clearly given up that it should be equal to this for the protection of the tetanus. Okay, so remember, it is not more than that on all that. It is written, if it is equal to 0.01, you will be protected. So answer to this question will be B. And in the tetanus, you know which type of immunization you give. You give a combined immunization also, you give a active immunization also with a toxoid, and you give a passive immunization with the HTIG. And we have already discussed them in the app and in the book also, that particular thing, that there are various categories at A, B, C, D, less than 5 years, 5 to 10 years, more than 10 years, and some fellows are there whose whose history you don't know, their unknown history. So what you will do regarding them, that we already discussed up. So remember here the answer is B, 0.01 IU per ml per liter. Okay, so this is the answer here. So that is not liter, that is per ml, 0.01 IU per ml. Fine. Now coming to this particular one, the diphtheria vaccine type is what? The diphtheria vaccine type, everybody knows that DT, that is a diphtheria is a toxoid. Diphtheria is what? Diphtheria is a toxoid. Diphtheria vaccine is a toxoid DT, and it can be used directly also, it can be used in combination also, DPT. Okay, and nowadays in the DPT also we are using acellular pertussis vaccine, and which acts as the adjuvant also. So diphtheria vaccine is a type of what? Toxoid. But if somebody asks what is the treatment of choice for diphtheria, the treatment of choice is ADS. And what is ADS? Anti-diphtheric serum, and it can goes from 20,000 to 1 lakh units as according to the severity of the disease. That will go this particular way. So what you are using up here, you are using a toxoid. As such, in the diphtheria vaccine type is toxoid, but anti-diphtheric serum is the treatment of choice. And up to 6 hours you can use what? You can use antibiotics. And antibiotics are which one? Like these penicillin and erythromycin. These can be used, but after 6 hours no use. Anti-diphtheric serum is there, which is mostly used up in the treatment. Because you know diphtheria is what? Diphtheria is a toxemia, not bacteremia. Okay, that you already know. Pseudomembrane formation and all that stuff is, you already know. Which of the following is the second line anti-tubercular drug? This everybody knows. It's very simple. Ethinomide. Ethinomide is the answer for this, and it is a direct forward question. Before it was HRZD, that is the first line, and in the second one, it goes the ethinomide and all. So you have number of other drugs like cyclosarin and all those kanamycin, capreomycin. Okay, nowadays new ones, linezolid is being used. Okay, bedaquiline and all that. So they all are second line ones, and the first line is HRZD. Now, a resident doctor developed needle prick injury, and what should be like the particular fellow from whom that fellow was taking a sample was HIV positive. What should be done and how much time? Okay, so answer to this question, what should be done is zidovudine, indinavir and lamivudine. This combination is to be given for 4 weeks. Okay, best is to be done within the 2 hours, and after 72 hours, no use. So this combination ZIL, and this was given up in my book also, that this combination is being used ZIL, zidovudine, indinavir, lamivudine for 4 weeks. And another option as per the latest protocol is what? You can use what? You can use tenofovir, you can use efavirenz, and you can use lamivudine as an option other than this for the post-exposure prophylaxis of HIV. Okay. Which is least correct regarding the Mycoplasma pneumonia, or which is not correct? That will be the better question. So answer is amoxiclav is not effective here. So this is not correct. Amoxiclav is effective. This is a wrong statement. What do you use here? You use macrolides. No organism is seen when cultured. Yes, no organism is seen when cultured. It is cultured, fine, on PPL agar and all, but you could not able to see the organism because it is not stained. Why it is not stained? Because it does not have a cell wall. Okay, walking pneumonia is known as, and bilateral infiltrates on the chest bronchogram you can get. Okay, on the chest X-ray you can get this bilateral infiltrate. So that is seen in this. Now, another question was regarding the chick sign, and over the nose and sides of the nose, also termed as what? Brownie nose sign. So this time they have asked chick sign. Next time they can ask brownie nose sign is seen in. So answer is chikungunya. It is a manifestation chikungunya, and this is a macular hyperpigmentation of the nose, and it is considered to be unique cutaneous manifestation of chikungunya fever. Okay, so chick sign is an important question. It is coming second time now, so you need to remember that chick sign, another name was a brownie nose sign, and seen in chikungunya. Now, as per the COVID-19 biomedical waste management ones, you need to remember certain things, that N95 mask which we wear up, head cover, shoe cover, linen gown, they are to be discarded where? In the yellow bag. Gauze, nitride gloves, latex gloves, face shields, they are there in the red bag. And viral transport media, Eppendorf tubes, pipette tips, they are also put up where? In the red bag. And this is also in the red bag. Okay, so this was a question in AIIMS, that where you will put the latex gloves and all. The answer is red bag. The only important thing to remember is that you have to use the 2 bags. Okay, why you are using 2 bags? To avoid the spill and all, tear and all. Okay, to ensure up that it should not drain up or it should not spill off. Okay, you should use 2 bags, and you should mark the bags with this particular label that it is COVID-19 biomedical waste. Okay, so that you have to do, and every particular institute is having its own protocol. Like some says all should be put in red, some says all should be put in yellow, but you go with CPCB and it shows like this, that yellow bag is for this, red bag is for this, and red bag is for these particular viral transport medias and all also. Okay. Now, this is asked, what is wrong with the Ebola virus? Which statement is wrong with the Ebola virus here? It belongs to this family, Filoviridae. That's fine. Mosquito transmission is not seen. How does it transmit? Contact with the secretion of the patient. Fine. And nosocomial infection, yes, it can be a nosocomial infection. And I've already told you direct contact. Which is the most virulent one? Zaire Ebola. Which is the least virulent? Ivory Coast. What is investigation of choice? PCR. Which levels are needed here? BSL-4. So these all things are regarding the Ebola. Fine. So this also you need to remember. So these are the various questions of the microbiology ones. Yes, I know that number of them might have the conflicts. 3 to 4 they might have the conflict. Rest of all are fine. And if you tell me the exact options, I can tell you the real answer of it. But up till there, I have given you a particular fundamental of it, that if it is this, then this will be the answer. If it is this, then this will be the answer. So don't worry that this was not the question, sir. How you have put this question and all that. You are telling this thing wrong. So don't play a blame game down. Just put up the options there, and I will love to tell the correct answer. I'm not saying I'm 100% correct and all. It's just a mixture of the things which I get from you only in the Facebook group of mine, and in the Telegram or in the WhatsApp group. This is from you only which I've got. I'm asking again and again what are the other choices. Nobody could able to tell up properly. So bear with me. Sorry guys if you feel like that this was told wrongly by the sir and all. This is my point of view regarding that question by knowing up those 2 options and all. I was not there in the exam, and I suppose you will able to feel up that thing, and I will say to you that if you have any relevant information regarding that particular option, you can tell up in the YouTube comment section, and we will get back to you and we'll tell you the right answer for sure. Okay, thank you so much, and prepare in and around these topics and all. That's for sure that nowadays they are talking about the history of the disease, and they are asking in and around. The coronavirus is a hot topic again in the November AIIMS. It will come, and obviously in the NEET exam the coronavirus will be full blown, will be there. Okay, you will get number of questions of coronavirus and all. So I will advise you to see up my video in my app also, the prep letter. Okay, you can download it from the Play Store and from Apple Store. And along with that, I will put this particular PDF also, which I have written up in and around this particular thing, into my group. Okay, that is on the Facebook and all, so you can see it from there. And I will give a link of that in the YouTube section also. Thank you so much, guys, and do tell me if at all you feel up that this was not there, this was there. I will love to hear from you. Thank you so much.